afternoon, everyone. My name is Sheena, and today I'm going to present to you upregulation of human glycolipid transfer protein, GLTP, induces necroptosis in colon cancer cells, colon carcinoma cells. And these are my authors. So I chose this paper because I used to work for a gastroenterologist, and we used to diagnose patients with colon cancer more often than what you would like to see. And then I also had some family members diagnosed with it, so I just thought it'd be nice to have more of an understanding if there's other ways to prevent it other than just a screening colonoscopy. And to discuss this paper talks about how GLTP overexpression can inhibit cell proliferation, and they do that by several different methods and procedures. And one of the methods that they used is basically arresting the cell cycle in the G1 phase at the checkpoint, and to see the expression then if cell proliferation was increased or decreased. I'm, however, only going to cover a particular topic in this presentation. I'm not going to cover the entire paper, so let's get started. What is colon cancer? So colon cancer is malignant cells in the colon tissue, and they usually begin as a polyp. And as we'll see here, these little circles are the tissues inside the colon, which is considered a polyp that will line the colon wall. And this particular is the transverse colon. And colon cancer is the third most common cancer in the United States, where annually there are more than 50,000 deaths that result from colon cancer, even with early detection and prevention. So some symptoms that one may experience is change in bowel habits, blood and stool, diarrhea, constipation, the feeling of not being able to empty completely, narrow stools, gas, pain, bloating, cramps, weight loss, fatigue, and vomiting. There are 5 different stages of colon cancer. They range from stage 0 to stage 4. And basically those stages increase by where the tumor has grown through the different layers. You have your mucosal layer, submucosa, muscle layer, serosa. Once it gets past the serosa layer, which is where stage 4 is, it starts metastasizing to other parts of the body, and then that's when, you know, a little bit more than just the surgery is going to help. So ways to diagnose and treat colon cancer. Colon cancer is diagnosed through rectal exams, fecal occult blood test, barium enemas, colonoscopies, and through the colonoscopy, if a polyp is found, then a biopsy is need to be done. And this is a picture of what a typical colonoscopy will look like. You have your physician and a patient with their backside exposed to the physician. A probe is inserted through the rectum, as you'll see here. At the end of the probe is a light and a camera. That light will allow the physician to see what's going on inside the colon, and then the camera will visualize onto the screen so the physician can look as he's navigating through the colon to see any polyps, any abnormal tissues, because if abnormal tissue of the colon is there, biopsies will be taken. And then if there are polyps, those polyps are removed through an instrument. The physician will then cauterize the area to stop any bleeding. And then other treatments would be barium enemas, chemotherapy, cryosurgery, radiofrequency ablation, and radiation. For this particular study, they looked at the human glycolipid transfer protein, GLTP. And this is an image of what GLTP looks like. It is a fairly small protein. It's only about 24 kilodaltons. It is encoded from chromosome 12, and that it is positioned on the long arm of chromosome 12 at the 20th position, 24.11 position. GLTP mediates non-vesicle trafficking of different glycosphingolipids, GSLs, and those GSLs, the glycosphingolipids, their particular role is surface adhesion, neurodegeneration, cell differentiation, drug resistance, and apoptosis. As you'll notice, GLTP is an all helical conformation, so there's no beta sheets. There's just alpha helical. And there is a 2-layer motif in the GLTP, which forms a sandwich type pocket. And this this is a pocket-like binding site for the glycolipid. So the glycolipid will fit inside and then is then moved throughout the cell. So the particular question that they used with GLTP in this study is, does overexpression of GLTP inhibit cell proliferation in human colon cancer cells? So as we see, they used 3 different cell lines. They used 2 colon cancer cell lines and 1 normal tissue cell line. The colon cancer cell lines are HT29 and HCT116. For this study, we're only going to see the results from the HT29 because the HCT116 was done in a previous study. And so if you was to read the paper, it talks about supplementary figures. And those supplementary figures would give the results from that previous study for the cell line of 116 of the colon cancer cells. This right here is an image of what the HT29 cells look like when they're under the microscope at low density. And then the normal cell lines is the CCD18CL. They then took vectors that they bought and purchased from a company, and they transfected those vectors into the cells. The vectors that they used was a flag tag as a control. This was the wild type GLTP. They then used a flag GLTP, which was basically the overexpressed GLTP from the previous studies, as I mentioned before. And then they also had a flag W96A GLTP. This had a mutated binding site within it. And that mutated binding site occurred because they went from a tryptophan at the 96th position to an alanine. And as we all know, if you change the structure of a protein, you also change the function. And then they decreased the expression of GLTP, and that was in a vector called the siGLTP. And then they used a vector called SI control, which was the non-targeting siRNA control. So to answer that question, they, as mentioned before, they transfected those vectors into the HT29 cell line along with the human normal colon cancer cells, along with the normal cell line. And that's what you'll see here, the HT29 and the normal colon cells. And what they saw was that the light blue stands for 24 hours, pink 48, and the dark blue is 72. Those is the time frame that they used post-transfection, that they looked and counted the cells to see if there was cell proliferation or was the cell proliferation inhibited. So using the control vector here, which is the wild type GLTP, they then used the overexpressed GLTP from the previous study, and what they found was that there was significant reduction in cell proliferation. And then when they looked and compared it to the normal cell line, there was no effect. And so they then were able to look at the flag GLTP with the mutated binding site, along with the decreased GLTP and the siRNA control vector. And they saw that there was no effect in the HT29 cells with those either. So therefore, the overexpressed GLTP had significantly reduced cell proliferation. It inhibited. So in conclusion and future direction, overexpression of human GLTP does induce apoptosis. And it does this through necroptosis. Necroptosis is unprogrammed cell death, where apoptosis is programmed cell death. And this only occurs in the human tissue 29 cancer cells. Cell death is regulated by sphingolipids, and that is through apoptosis. An example of the sphingolipid would be a ceramide. Then for future direction, even though much is known of the structure and function of GLTP, and in vivo functions of SL transfer proteins is still unknown. So more needs to be done on that particular area. And then expressional relationship between GLTP and FAPP2, which is also another protein, impact on sphingolipid metabolism still needs to be evaluated. So that still also is another future question as well. And these are my references. Thank you.